Hi everyone, today we are doing lecture number 5, Energy Concepts. So this lecture is a little bit of a segue to give you guys some background into some of the basics behind energy before we get into the specifics of the cellular metabolism lecture. So we don't really have any textbook references for you for this particular lecture. It's just a little bit of background. So we're going to talk about the basics of energy. What is energy? Some of the laws of thermodynamics behind energy. Talk about the specifics of ATP and how ATP stores energy. And then we're going to get into enzymes, enzymes which help chemical reactions take place if they are highly energetic reactions that need assistance. And then this will segue us into biochemical pathways for the next lecture, where we're going to talk about cellular respiration and metabolism. Okay, so energy is the ability to do work or to put matter into motion. So we talked all about chemistry in the previous chemistry lecture. And so energy is really the dynamics behind how things do work. So energy is present in the universe in many forms. Some examples of energy. Energy is present in heat. Energy can be in the form of light waves. Energy can be in the form of sound or sound waves, which is really kind of a vibration. Energy can be in the form of electrical energy, which is the movement of electrons. Mechanical energy, which is actual physical motion. So we can put energy into something, and when it moves, that is mechanical energy. And then chemical energy. And a lot of what we're going to talk about in the next couple of lectures is chemical energy. And that is the energy that is stored in chemical bonds. For example, the energy that is stored in the phosphate bonds of ATP. So energy flow, I love calling it energy flow. This is kind of, I feel like we're taking a yoga class, right? Energy flow. It all flows. Really, we're talking about the movement or the change in energy as we go in different forms, between different forms of energy. So energy in terms of our world flows from the sun to the earth. So the energy from the sunlight or the light waves is captured by plants and stored in carbohydrate molecules. If you ever have a chance to take a plant biology course and look at the process of photosynthesis, it really is a fascinating and amazing biological process. And that is the process of taking energy from the sun or sunlight and storing it in carbohydrate molecules or carbon bonds. So us as humans and animals, we then will take carbohydrate molecules and other macromolecules like lipids and proteins, and we can use the energy in those bonds, the carbon bonds in organic molecules, to make another energy storage molecule, ATP. So in our bodies, energy is stored in macromolecules like carbohydrates, lipids, and proteins, and in the form of phosphate bonds in ATP. So in the biological world, food energy passes through an ecosystem. So plants, algae, and bacteria, some bacteria, can obtain energy from the sunlight and produce energy-rich carbohydrate or sugar molecules, right? Sugar is the common name for some carbohydrates. So all other organisms that cannot capture energy from sunlight, all other organisms are going to be in these categories. They will be consumers, and they will have to obtain energy by consuming either the primary producers that got energy from the sun, meaning plants, or by consuming other consumers and converting the molecules in those organisms, plants or other animals, into energy. So we get carbohydrates, proteins, lipids or fats from the food that we eat, and we then convert those molecules into energy. So again, some energy forms. Chemical energy is potential energy stored in chemical bonds. Primarily in physiology, we'll talk about energy stored in carbohydrate bonds. Energy stored in carbon bonds, such as in carbohydrates. Energy stored in phosphate bonds, such as in ATP. And so these will be the typical bonds that you'll see in food. Those will be the carbon bonds. And the typical bonds that you will see in energy sources like ATP. That is all chemical energy. Mechanical energy is energy directly involved in moving something, for example, muscle movement or muscle contraction. So we will take energy from some other source, like, say, the chemical bonds of ATP, and we will use that energy to create movement, and that's mechanical energy. We also have electrical energy, and this is energy that flows within charged particles or ions. We'll talk a lot about this when we talk about the nervous system, because the nervous system uses the movement of charged ions, sodium, potassium, and others, as electrical energy. There's also radiant energy, and this is energy that travels in waves. So, for example, waves of sunlight. So, the sun provides a type of radiant energy, which is sunlight, to plants. Plants use the process of photosynthesis to store that radiant energy now as chemical energy in the form of molecules like carbohydrates. Animals, including us, will then take the energy in those carbon-based molecules like carbohydrates and break it down in the process of cellular respiration to make energy which we can store in our bodies. We then have energy to do work. There are some byproducts of this reaction. These reactions, water and carbon dioxide, are byproducts of cellular respiration in animals. Oxygen is required as input for human respiration or general animal respiration. And for photosynthesis, carbon dioxide is required as input for photosynthesis. Some energy will be lost in the process to the environment. So there is energy lost in the transfer between different forms of energy. So the basics behind this energy dynamics is thermodynamics. So thermodynamics is a branch of physics which describes the relationship between heat, or other words, thermo energy, and other types of energy. So there are two basic laws of thermodynamics which describe the basic concepts of energy in the physical world. There's the first law and the second law. Thank you for naming them in some way instead of after a person, right? So the first law of thermodynamics says this. Here's the formal law. The total amount of energy in the universe is constant. Energy is not created or destroyed. It is only converted from one form to another. So say chemical energy to mechanical energy, etc. From this, we know that energy is always present and will be actively used or stored. So energy does not go away. It's converted from one form to another, or it is being stored as potential energy. So when energy is actively being used or doing work, you guys ever see that series Robin Big? That was a great TV series, Robin Big, and they used to always say, do work. That's what I always think about when we say doing work. So energy is the ability to do work, and is the energy of that is actually currently doing work, or the energy of motion or currently in use. And this we call kinetic energy. So kinetic means movement. The opposite of that would be potential energy, and this is energy that is not currently being used, but it is not destroyed, it doesn't go away, it's just being stored. So we're holding on to it. It's energy in the bank, and that's potential energy. So here's the difference between potential and kinetic energy. So potential energy is when you are, for example, holding on to a ball at the top of a hill. So we know that if you let go of the ball, that it has the ability to roll down the hill. So it has the ability to do work, and that's potential energy. If you let go of the ball and the ball rolls down the hill, then the ball is now actually moving. This energy is doing work, and that's kinetic energy. So potential energy is energy in the bank that we're saving up, being stored. Kinetic energy is energy that is actually actively being used. Okay, the second law of thermodynamics deals with heat and something we call entropy. So heat is a product of energy conversion from one form to another. So during these kind of reactions or conversions, you will see heat often being released as energy is converted. And in addition to that, entropy is the amount of disorder or chaos present in a system. So basically what entropy tells us is how likely is it that something will occur. So most systems will degrade and release energy. This is the natural state of things. So conversions in energy will produce entropy. The flip side of that is to say that organization or structure, holding things together, requires an input of energy. So I think of this like I think of cleaning my room. It's much easier for my room to get messy as I sort of go about with my daily activities, and that sort of chaos releases energy. That sort of flow of the normal everyday is total disorganization, right? So left alone, most systems will degrade and release energy. It then takes energy input to organize and clean my room, right? So structure, putting things back together or maintaining things together, requires energy input. So in any cyclic process, the entropy is more likely to increase or become more disorganized than it is to decrease. It takes energy to decrease entropy or make something more organized. There is the possibility also that entropy can stay the same. So no change is also okay. But more likely, entropy or disorganization will increase. And this is the second law of thermodynamics. So let's say this in a couple of different ways because I find that entropy is a bit of a challenging concept. So living organisms, as it relates to physiology, they are organized, right? They have low entropy. In order to maintain low entropy, a constant input of energy is needed into the body of the organism. For plants and other producers in the ecosystem, sunlight energy is required to sustain life. So they need light to make energy in the form of, say, carbohydrate bonds required to sustain life. For humans and other animals, we need food energy either from plants or other animals to obtain energy for our bodies to maintain low entropy. So this is why we need to eat and obtain food constantly, because we need a constant input of energy into the body of the organism to maintain this low entropy state. It takes energy to maintain an organized state. So energy conversions in humans, here's an example. So we will take in food and we will absorb the food that we eat and store carbohydrates, lipids, and proteins from that food. A calorie is a unit of measurement of energy stored in the food. We will then use that chemical energy stored in these food molecules to produce ATP. Cellular respiration, which we're going to spend the entire next lecture on, is the process of making this ATP in our cells. ATP has high chemical energy storage in its phosphate bonds, and it is the main energy currency, or the money we have in the bank for our bodies. So here's another way of looking at it. This is fuel consumption or energy conversion in a car, which is kind of similar to the fuel consumption or energy conversion in our bodies. So cars need energy, chemical energy in the form of gasoline. We add that to oxygen, and through the process of combustion, we can then turn that energy into kinetic energy to move the car. Some heat will be released in that transfer process. In addition, waste products, carbon dioxide and water, will also be released. In our bodies, the fuel that we use is the food, carbohydrates, lipids, proteins. We combine that with oxygen in the process of cellular respiration. Cellular respiration converts those food molecules, or the energy in the carbon bonds, into energy in phosphate bonds of ATP. And then we have energy stored in the bank, in the cell, in order to do cellular work. As that transfer takes place, there will also be heat energy released. And the byproducts of that will be carbon dioxide and water. More specifics on the cellular respiration chemistry in the next lecture. I know, don't get too excited. Okay. So calculating calories is one way to look at food energy conversions during the metabolic processes of the body. So a calorie is the amount of heat energy used to raise 1 gram of water 1 degree Celsius. Now, this is the formal definition of a calorie. Calorimetry is the measure of heat energy released from the body through metabolism. So we can actually measure, that's the metric part of the calorimetry, the change in heat or the change in oxygen during metabolism. Change in temperature is the most direct measure of calorimetry. So when the energy is converted from food into energy, then heat will be released as a direct product of that conversion. That change in temperature can be the most direct measure of calorie changes, but that is very difficult to measure in a laboratory setting. Easier to measure in a laboratory setting is the byproduct, which is one of the reactants, which is oxygen. So as we break down food molecules in the process of metabolism, we use oxygen to do that. So we can measure indirectly the amount of oxygen in a closed setting. And as the oxygen gets used up, that will then be an indirect measure of how much was used to create energy during metabolism. We're going to do this in the lab during the metabolism lab, so more on that later in lab. Okay. Metabolic rate then is the total energy expenditure per unit time. We will calculate this in kcals per hour using the average time for oxygen consumption at room temperature and standard air pressure. Okay. All right, so on to ATP. So ATP is the energy currency used by cells in our bodies to do work. It is chemical energy that is produced in mitochondria. So go back to our cellular lecture. Mitochondria are the powerhouse of the cell. So mitochondria are actually responsible for producing ATP during the process of cellular respiration. This process requires food, oxygen, several enzymes and biochemical pathways, and it will cause byproducts. It will release carbon dioxide, water, as it forms ATP. So I want you guys to memorize this equation, and we will go through in great detail, more detail than you ever wanted to know, next lecture. So here is a sugar molecule, C6H12O6. We combine that with oxygen, so this is food plus oxygen, through the many biochemical processes of cellular respiration, and we get ATP. The byproducts then will be water and carbon dioxide. So here's the structure of ATP. ATP is made up of a nucleotide, which is adenine, a sugar, which is ribose, and 3 phosphates. And it's the phosphates that are really key for ATP. So here's adenine, which is the nucleotide base. Here's ribose, which is the sugar. And here are 1, 2, 3 phosphate groups. So it is the phosphate bonds in ATP that are high energy. So think of them like coiled springs. They are storing energy within their bonds, and when we release that energy or break the chemical bonds, then we get energy released that can be used in a cell to do work. So energy is stored in the chemical bonds of ATP as chemical energy in the form of phosphate bonds. It'll be released from ATP every time a phosphate bond is broken. This forms ADP. So ATP, the T is for triphosphate. When we break off one phosphate group or one phosphate bond, this forms a DP or diphosphate for 2 phosphates. If we then break off the second phosphate bond, we're left with 1 phosphate or monophosphate, AMP. Typically, we just go from ATP to ADP though, and so this is a summary of that reaction. So when a cell needs energy, it's got ATP in the bank. So it's going to go to the ATP bank, it's going to break down ATP to ADP and release a phosphate. When it does that, energy will be released into the cell, and then the cell can do work. So ATP will be used and recycled as needed to store energy or release energy. So cells constantly need energy in the form of ATP. Cells are very dynamic. They are constantly doing work. They're making proteins. They are recycling parts of cells. They are doing active membrane transport. Some cells are contracting or moving, like in the case of muscle cells that need a lot of ATP. And again, synthesis of molecules. So energetic processes in cells can be any of these examples. So there is chemical work, which is doing chemical reactions, making or synthesizing molecules, making products, right? We spent a whole lecture on the protein synthesis. So for example, forming those peptide bonds, that's going to require energy to make proteins. We also spent an entire lecture on membrane transport, and some membrane transport, which ones? Think about them. The active transport category, things like what? Primary, secondary active transport, endocytosis, exocytosis. Those processes require ATP, either directly or indirectly, to do transport work. That is moving molecules from one side of the membrane to another. There's also mechanical work, for example, in muscles, as the proteins of muscles move to contract muscles. They require energy input in the form of ATP. And that's mechanical work. So metabolism then is the sum of all chemical reactions that occur in the body, and there are two types of metabolic reactions. There are anabolic reactions that use energy to synthesize large molecules, and there are catabolic reactions that break down molecules to release energy. Usually, that energy is stored in the form of ATP. So let's just review a bit about chemical reactions to move you guys towards our cellular respiration lecture. So a chemical reaction is the making or breaking of chemical bonds to change. We're either going to form or synthesize, or we're going to break down molecules. They are described in this basic form. We have a reactant, reactant 1, plus reactant 2, and that goes to a product. A reaction is more likely to occur if it releases energy than if it requires energy. So given any reaction in the body, we generally want to ask, how does this reaction happen and in which direction will it go? So if it requires energy input, then it's called an endergonic reaction. If it releases energy, it's an exergonic reaction. If you think back to chemistry, these were sort of the cold versus hot type reactions. So if you have a reaction that releases energy, that will often actually be felt in the form of heat as that reaction is taking place. So chemical reactions in the body include these two basic categories, synthesis and decomposition. So synthesis reactions are atoms or molecules are combined to form larger molecules. So A plus B forms the combination AB. This type of reaction typically absorbs and stores energy in the chemical bonds that are formed. So that's an endergonic type reaction. So for example, amino acids will join with peptide bonds to form a protein, and that requires energy. The flip side of that would be decomposition reactions. And this is when large molecules are broken down into smaller molecules. So you start with large molecule XY and you break it down into its components X plus Y. This type of reaction is breaking bonds. And when we break chemical bonds, then we release the energy that is stored in those chemical bonds. So that will release chemical energy as chemical bonds are broken, and that is an exergonic reaction where you will also often be able to see or measure the heat that is released alongside that breaking of chemical bonds. For example, glycogen is what is it? It's a polysaccharide, so a large carbohydrate that's made up of many glucose rings bonded together. Glycogen will be broken down in a decomposition reaction into several glucose molecules. So here's another way of looking at endergonic versus exergonic reactions. So an endergonic reaction requires energy input in order to take place. An exergonic reaction, however, releases energy. So if we look at the amount of energy that must be put in, an endergonic reaction is like climbing up a hill. You need to add energy, which we call activation energy, in order for that reaction to take place. An exergonic reaction is like going downhill. Energy will be released, and it's very easy to do an exergonic reaction. Which one of these has more entropy? The exergonic reaction is more likely to take place because it has more entropy, right? Okay. So all chemical reactions are theoretically reversible, and they will be written with arrows that go in either direction. So let's go back to our previous one. Notice that there are double arrows going in either direction. So from reactants to products, A plus B goes to AB. We can also do the flip side, which will be the decomposition. AB goes back to A plus B. So reactions are always reversible, that doesn't mean that they're actually going to take place in any biologically relevant time frame, though, right? So if it's going to happen 1,000 years from now, then we'll be dead by the time it takes place, right? So theoretically, they can be reversible, and the direction will shift depending on the concentration of reactants and products. So remember, back to chemistry, if you add more of A or you add more of B, then that will drive the reaction towards the right. Okay? It'll move the reaction towards the formation of AB product. On the other hand, if you add more of the product or you add more of AB, this will move the reaction to the left, and it will drive the reaction in the opposite way to break down AB into A plus B. So the actual likelihood of the direction will also depend on the energy it takes to form or break the bonds in the reaction. So if it takes a lot of energy input to form the products of a particular reaction, then it is less likely to occur. So how do we do this in the body if we need these products to be formed, but it takes a lot of energy to do so? So this energy is called activation energy, and I think of this as, I always say this to my husband all the time, right? You're sitting on the couch, you're watching TV, you really should go do something, but you just need that push. That push to get you off the couch to go do some work or maybe go work out is activation energy. So you need some activation energy to get started. Enzymes are proteins that will help with this problem. Wouldn't it be nice if we had like a large version of enzymes to help us get off the couch? Anyway, so enzymes are proteins that make reactions faster. So they do this by binding to the reactants, changing their shape, and decreasing the activation energy required for the reaction. So in other words, reactions that might normally be very, very slow will be increased and made faster by the presence of enzymes. So it's like your friend that comes over and says, hey, get off the couch, right? That's what the enzyme does. So enzyme action is called catalysis, and a reaction is made faster or catalyzed by an enzyme. So the enzyme gives it that extra push to get the reaction started. So in other words, enzymes reduce the activation energy required to get started. So without an enzyme, going from reactant to products would take a lot of activation energy. So from here to here, that would be without an enzyme. That would be uncatalyzed. But with an enzyme, that activation energy gets significantly smaller, and the reaction is more likely to take place. So here's one specific example of reaction rates that are faster with enzymes. So here is a substrate, glucose, going to a product, glucose 6-phosphate. Normally, the rate of that would be 0.000000001 micromoles per liter per minute. That is super slow, right? But with the enzyme hexokinase, this reaction now is at 1,300 micromoles per liter per minute. That is a 13 billion time acceleration because of the decrease in activation energy that was performed by the enzyme. So enzymes will bind to reactions specifically like a lock for a key because they have a unique three-dimensional shape. So what kind of macromolecule is an enzyme? Think back to your macromolecules lecture. We talked a little bit about enzymes there, and we're going to add more details here. They're proteins. So an enzyme is a protein. So it's made up of many types of amino acids. So each of these little circles here is an amino acid. They're not different colors, although there are many types there. And proteins including enzymes, have very specific three-dimensional shapes. So this is the tertiary folding of a protein, for example, an enzyme. And the folding of an enzyme is very specific to create an active site in which the reactant can bind. These active sites are specific for a particular reactant. When the reactant binds to the active site is not changed. The reactant can go through with its reaction, leave, and then the enzyme can be used again with the active site in the same position. So enzymes work by bringing reactants closer together. So I use the example of kindergartners running around a playground, right? You're trying to get them all lined up and you're trying to get them to do something. What an enzyme will do is grab one, grab another, put them together, okay? So normally the reaction could take place, right? If it's just out, but it might not be very likely to happen. But if you take one, you take the other, you put them together, then you have a reaction, and that reduces the activation energy. So here is an example of an enzyme where the active site has reactant 1 and reactant 2 binding sites. The reactant 1, reactant 2 will come together for this enzyme substrate complex. Now they're close together. Now it's much more likely that this reaction can occur. The activation energy is lower, and we can very easily form the product, which is the combination of the two. After the product is formed, the enzyme is unharmed. It can go back and find some more reactants and do it again. So some enzymes will need to be assisted to change to an active shape to bind to their reactants. If an enzyme needs another molecule to help it have a more available active site, that molecule is called an activator. Often the activator will bind to a separate site on the enzyme. It won't bind to the active site, it'll bind somewhere else. And that separate site is called an allosteric site. An enzyme can also be inhibited if another molecule changes its activity. So in the case of an activator will increase the active site availability, make the active site even more active. In the case of an inhibitor will actually decrease the active site availability. And there's two ways to do that. You can directly block the active site, and that's called competitive inhibition. And that is when another molecule binds to and blocks the site directly. Often this is like a substrate imposter. So it's something that looks like a reactant but isn't actually the reactant, and it will bind to the active site because they look and have similar shape, but it'll block that active site so the reactants can't get in. The other example is when another molecule will bind to a separate location. So you've got your enzyme with the active site. The other molecule will bind somewhere else away from the active site, but in doing so changes the shape of the protein, changes the shape of the enzyme, and the active site will no longer be available. This is non-competitive inhibition. It's a little bit more, say, passive-aggressive. It's not directly blocking the active site, but it can cause the active site to change, so it's no longer available. So let's look at some examples of using these allosteric or opposite sites to activate or inhibit an enzyme. So here's our substrate. And the substrate normally, when the enzyme is active, will be, in this example, broken down into products. So the substrate goes into the enzyme, and then the reaction takes place. It'll be broken down into products. In the case of an activator, normally the substrate doesn't bind very well, but you put in an activator in a separate site. So here's the active site here, here's the separate site. You put an activator into the separate site and it makes that active site better. Now the active site binds very well to the substrate and the products can be formed. In the case of a repressor, the substrate normally would be binding. The repressor will come in to the separate site. So here's the active site down here. The repressor comes into the separate site and changes the shape of the molecule, of the enzyme, such that the substrate no longer fits in that active site. And now this enzyme cannot help the reaction take place. So it's been repressed or inhibited. So here's a view of competitive and non-competitive inhibition. So the one that we just talked about was non-competitive. So that is where you have the enzyme being inhibited by a molecule separate from its active site. So here's the active site here. The substrate can bind to that active site. But if the non-competitive inhibitor is bound separately at this other site, the other site is called allosteric, then that messes up the active site. The substrate can no longer bind. That's non-competitive inhibition. A competitive inhibitor will directly get in the way of the substrate. It'll bind to the actual active site of the enzyme and the substrate is blocked. So this is where I like to say, guys, you're out at a bar, you're trying to talk to a lady, another guy comes and sits next to you, and he starts talking to the lady, kind of competitively inhibiting. There's a word for that, right? It's a certain kind of a blocker. I know, just trying to keep you guys awake. So competitive inhibition is a direct blocking of the substrate to the enzyme, a direct blocking of the interaction itself. A non-competitive inhibitor is somebody from across the room that prevents the binding of the substrate in another way. Okay? Sometimes the product of reaction is a non-competitive inhibitor in order to limit the total amount of reaction taking place. So sometimes the inhibitor is actually the product. So this is another layer of complexity that is basically feedback inhibition. So the accumulation of the end product then will come back to inhibit the enzyme. And what this does is, as the product is being formed, it will come back and tell the enzyme, hey, we have a lot of product that has just been made. You can shut down now. We've had enough activity. So this type of feedback inhibition, where the product feeds back and inhibits the enzyme that created it, will turn off or slow down a metabolic pathway so that things don't domino and you don't make too much product. There are other factors that affect enzymes, and these are generally factors that affect chemical reactivity. One will be the concentration of a substrate or an enzyme. So more concentration, more substrate, more enzyme will increase the reaction rate. Another is temperature. Increased temperature increases kinetic energy of molecules, and that increases the reaction rate. But remember back to our macromolecule lecture, and proteins, including enzymes, will denature at high temperatures. They will lose their folding, they will lose their shape, and if an enzyme loses its folding, the active site is no longer binding, right? The last is pH. So most enzymes have an optimal pH range in which their shape or conformation is best. Again, if the pH is too acidic or too low, the enzyme can also denature. Okay, did you notice these are also homeostasis factors? Concentration, or the level of fluids, electrolytes, the level of molecules in the body. These are things that our body is constantly regulating. Temperature is constantly being regulated. pH is constantly being regulated by the body. So a lot of this basic homeostasis comes back to being able to regulate the chemical reactions in the body. So here's an example of enzymes being sensitive to their environment. So this is another way of saying that temperature and pH affects the activity of enzymes. So if we look at a human enzyme that's normally most active inside the human body, the optimum temperature for that enzyme is very close to human body temperature, 37 degrees or so. If we get too high, then that enzyme will no longer work. For bacteria or enzymes that are normally present in bacteria that live in hot springs, these are organisms that are living at much higher temperatures. Their enzymes are actually more active at the temperature at which those organisms live, and they will trail off at either end based on the environment that they are used to. There's also optimum pH for different enzymes in the body based on locations in the body. So pH for an enzyme called pepsin is very close to the pH at which this enzyme lives, which is in the stomach. Very acidic in the stomach. This enzyme does very well in acidic environments. This enzyme trypsin is present in the small intestine, which is a more balanced or more alkaline pH compared to the acid of the stomach, closer to pH 6 of 7. That's the optimum pH for trypsin, which is located in the small intestine. So this all segues us, and hopefully this lecture helps you guys to understand a little bit behind the energy concepts for biochemical pathways. So in biology, some end products will require a series of these types of chemical reactions and enzymes. Often, different organelles in which these biochemical pathways occur will have compartments for these enzymes and chemical reactions to be located. So a biochemical pathway is a series of reactions necessary to make biological products needed for a given cellular process. And we're going to talk about the mother of all biochemical pathways when we talk next lecture about cellular respiration. So we will talk about how these biochemical pathways will be a series of catalyzed reactions occurring together in sequence. So in this case, the product of one reaction will be the substrate for the next reaction. Then that reaction's product will be the substrate for the next reaction, so on, until we get our final product through that series of biochemical reactions. So a series of reactions occurring together where one supplies the other is a biochemical pathway. And we will talk about in the next lecture the cellular respiration pathways in which we take carbohydrates and convert them into energy. Okay. So thank you for your patience today. I will see you for the next lecture, cellular respiration.